name's Charlie Kepler. I work here at Headwall Photonics in our customer support division. I'm also one of our lead pilots and do a lot of applications engineering as well.
I'm Carson Roberts. I'm a principal applications engineer at Headwall Photonics. I try and use our systems the way that our customers might. I spend a lot of time going out into the field and collecting data, trying to find new applications for the technology. Headwall is a manufacturer of components and systems based on diffraction gratings. We make single point spectrometers, we make spectrometers to go into other people's original equipment. We sell diffraction gratings themselves, and my part of the business is we use those diffraction gratings to make hyperspectral imagers, capture imagery with them, and do spectroscopic analysis on the spectral imagery.
This mission we wanted to, kind of as a proof of concept, take the nano hyperspec instrument into the field and do some ground truth scans with the tanks that had the coral inside that they were rehabbing to go be outplanted back into the ocean, and then take that same instrument and put it onto the M300 UAV and fly over the ocean where they have recently done some of these outplanting events to see, can we see what they have outplanted? Is the coral healthy?
My name is Marian Biddle. I'm a staff biologist at the coral restoration team here at Mote. I see to our point. Working with the coral restoration, so anything from fragging to daily husbandry tasks of algae mitigation and siphoning to coral feeding.
These are the guys that are still kicking, and some of them doing surprisingly well in wake of the heat wave last summer.
I hope that this mission results in a real-world application that we can use hyperspectral imaging for determining coral health.
Hyperspectral imaging is potentially useful in any situation where there is a scene, where there are objects in the scene that have characteristics that show up as spectral features. For example, I'm surrounded by the green grass. That's a result of the chlorophyll in the grass, which is a chemical which absorbs and reflects light. And so there's a lot of people that have been using hyperspectral imaging to look at plant health. Essentially, by looking at the spectrum of a plant, you can get an idea of how much chlorophyll it has. The more chlorophyll plant has in general, the healthier it is.
Hyperspectral imaging can help companies like Mote in kind of seeing things that the naked eye can't really see, detecting things early, differentiating different species, telling healthy versus diseased, maybe to catch something before it becomes a wider spread issue.
I grew up snorkeling and appreciating the reefs in the Caribbean and here in the Florida Keys. And so it's really important to me that we protect our valuable resources.
Well, eventually is to have the ecosystem at the previously healthy state it was, and it to be able to sustain itself.
A lot of minerals will have absorption features in the shortwave infrared, and we can use our shortwave infrared hyperspectral imagers to take pictures of either mined areas or natural scenes and identify the minerals present. Another very interesting thing that we've done with the hyperspectral imaging recently is we've been able to identify multiple different species of toxic cyanobacteria in ponds. And again, this is a way of solving a broader problem of the harmful algal blooms and quantifying them, again, by remote sensing without having to send people into the field to sample the water.
I went to undergrad for mechanical engineering, and I kind of on a whim started working at Headwall as a project manager out of school, kind of as a blind cold call from a recruiter. And ever since then, I kind of have worked into the remote sensing area of the business and am involved in all of our drone flying and things like that.
These have been recently rescued from either Opal Key Rescue or the Coast Guard Rescue.
And that's because they're doing construction?
Yeah, so there was some construction, and because we're in a part of Florida Keys National Marine Sanctuary, any construction or manipulation to the marine environment or potentially harming any of those sensitive marine resources have to be either mitigated, like if there is harm, so you'll have to pay for the potential harm, and or rescued.
I left high school determined to be a physicist. I went to school for a very long time and got a doctorate in low temperature semiconductor physics, and then have had a wide variety of jobs in the time since I got my degree. And I sort of fell into hyperspectral imaging through a colleague I knew from grad school who was working on this technology and needed some help, and that was 12 years ago.
When we outplant these plugs here, we space them in about clusters of 10 of all one genotype, and once they grow out and grow onto and close to one another, they will then merge and form about a coral of this size. And once they get to this size, they're then sexually mature, and then they can reproduce.
Any coral that's been on land for more than 30 days has to be vet checked by a professional before it goes back out to the ocean. It's, like I said, it's a PVC structure with fiberglass rods for the branches, and duckbills are hammered into the bottom of the ocean, and then roping, these things are tied down and they float in the middle of the water. The reason why it's such an effective strategy is because you're essentially hanging these corals in the middle of the water column. So you're above all of the detritus and stuff on the bottom, you're not too close to the top, and you're maximizing on both sunlight and the nutrients that come by for their feeding, and then they stay there until they grow to entire plug size, and then those will get outplanted as well.
Hyperspectral imaging can be used for really anything. Everything reflects light at different wavelengths and different intensities, and so you can tell the difference between a tree forest and camouflage. And so it can be used in mining, it can be used in defense applications, agricultural, forestry, really kind of just across the board. There's kind of an opening over there that might be a better spot.
We frag the coral from that big size into little smaller pieces, and then we can make like hundreds of coral within 4 to 6 months instead of 40 or 50 years to get to that size. We cut them ideally anywhere from like 1 centimeter by 1 centimeter square frags to larger frags, and we're still in the experimental process to figure out the expedited growth for those species.
These operations will need to scale incredibly in order to make a real impact on the environment. And it's not going to be possible for individuals handling individual samples to restore the coral reefs. It's going to have to be a big industrial scale operation, and this is exactly the sort of thing that hyperspectral imagers can really help with.
So once they grow out to about 3/4 to a full-size plug like this or like this, they're then ready to be outplanted, at which point we have them vet checked. So we have a coral vet do a health assessment on them, and once they're approved, they then can be outplanted on the reef here.
So I'm not just looking at how our corals are doing over time. I'm taking a look at sponge communities, soft corals, the pre-existing coral communities. So what you're seeing right here is our baseline and then 1 year post-outplanting. So this was taken pre-outplanting, but here are our 1-year-old, our aquaporas cervicornis. We use Nikon DSLRs. We have the D7500 and the D7000. So each of those dots is a photo.
Southwest quad, 30%, minus 50.
I was originally a marine science and biology major at the University of Miami. My sophomore year, I took an invertebrate zoology course, and I got really interested in that professor's work. She runs the Cnidarian Immunology Lab up at UM, and I was able to work with a PhD student, and that's kind of how I got into the coral world. My thesis was actually on disease and microbiome stuff with coral. I was able to do an internship here, got introduced to the ecology side of things.
My name is Celia Leto, and I'm the Coral Reproduction Laboratory Manager at Mote Summerland Key Laboratory. I oversee all sexual propagation of coral at our facility. Our main facility is located in Sarasota, Florida. That's where you'll find our aquarium and a lot of our research. And then we are located in Summerland Key, Florida, which is about a half an hour north of Key West. We house our Coral Health and Disease Program, which is Reproduction Lab is a part of. We have our restoration team, ocean acidification team, our marine operations team, and then our ecosystems monitoring team, as well as our microbiology team. They're a part of Coral Health and Disease, but it's a big conglomerate. Our focus is on researching all aspects of the coral. One of our main goals, specifically in relation to reproduction, is increasing genetic diversity by allowing the reproductive process to happen in a controlled setting. So the reason I essentially have a job is because we are unfortunately observing an almost complete failure of natural reproduction in the wild. So it is my job to make sure that we are not only raising corals that are large enough and healthy enough to be able to reproduce, but to study them and optimize how we can continually improve upon this process.
We had a volunteer program of combat wounded vets come down here 3 weeks ago, and they helped us do a bunch of coral fragments. Yeah, it was a really fun program.
How do you actually split them up like that?
So, it's very complicated. We take a bandsaw and we move them through them. It's a diamond bladed saw, so it's friction based, so like you can nick your fingers on it and it won't cut skin or anything. So we just move them through that saw, kind of like hijacking their asexual reproduction, because corals, as I mentioned before, they're capable of sexual reproduction, spawning stuff we want them to do. But the way that they grow larger naturally is asexual reproduction as well, budding and lateral growth as the skeleton grows under them. Corals that are physically abrased in some way have an enhanced growback period.
These are some important individuals because some of these genotypes are actually no longer found in the wild, so they're also for gene banking purposes. And this system is artificial saltwater. It's programmable and it's recirculating, so that means that we're isolated from natural saltwater, so we don't have to worry about a lot of the things that can happen in the ocean that we don't have control over, like rising temperatures, oceanification, disease, etc. And then we can control their temperature seasonal cycle, the solar and lunar cues as well. And that's what they use to synchronize their spawning behavior. They will release gametes or egg and sperm into the water column once a year, usually around the full moon in August in the middle of the night, which is not fun for me, but it's great for them, using those cues. So every year I will program whatever conditions we want them to experience, and then they will spawn in captivity. And it's really special because we are able to complete a coral entire life cycle in-house, which is something that makes our institution and our laboratory unique. 2023, we experienced the worst thermal event that the forest coral reef has ever experienced yet. And we were really worried about how our outplanted recruits would do, of course, because I'm a mother and I want my babies to do well, but also just what that means for the environment as a whole. Fortunately, at least our specific area and a lot of different areas throughout the Florida Keys did experience a large amount of loss, specifically in aquaporids, so staghorn and elk-pro corals and the branching species, because they are rather susceptible to thermal stress. So in our monitoring, of course, there were all these sad results coming back from multiple institutions, but we found that many of the outplant sites that contained our 2020 and 2021 cohorts of Aquaphora silvercornis and Flamana had upwards of 70% individual survival and 100% familial survival, which is amazing, not only just out in general, but it's also great considering the terrible conditions that they were in. And that's showing that by strategically crossing specific parents that we know have desirable traits and making sure that we are diversifying that population, that we are increasing that adaptive potential. And that is real world evidence that those guys survived. Some of them never even bleached, and they're growing really well still to this day. So that means that what we're doing is working, and now we just need to focus on optimizing and upscaling that so that we can match the ever increasing issue. When I was 13, I asked for a scuba diving license for my birthday, and my parents were nice enough to let me. So I was certified, I went diving, and then I was able to actually go scuba diving in Coral Restoration Foundation's original infield nursery. I was able to do a high school capstone with them, where I just essentially went out diving and wrote a paper on what they did. But that was the first time that I had really heard about what was happening to our coral reefs and how they were declining at a very alarming rate, and how there was something that you could do, and that it was a real job. So after I learned that, I just decided that's what I wanted to do. But I ended up going to school at the University of Miami. I earned a dual bachelor's in marine science and biology, and that really showed me how many different areas that we can research and how much we don't know yet, and it was really exciting to me. Corals are very, very small, very tiny, and very hard to find on the reef. And although we know that they're not really recruiting at good rates, we do know that there are some locations that corals are still able to reproduce with those as spawning hubs. How effective those spawning hubs are is kind of under-researched. And the reason is because it's so hard to find these little babies, and it really depends on what time of year that you go out. So I imagine hyperspectral imagery would be a fantastic way to highlight where those recruits are.
What are you mixing up? Oh, it's in the food right now. It's called golden pearls. It's more or less just like crushed up oyster eggs and stuff.
For the coral?
Yeah, the coral.
Keeping track of genetic diversity in our lab and in our various nurseries and where those individuals end up is a very large task. And currently we are using a combination of our partners with Seafoundry for coral restoration efforts, and then for our sexual propagation needs, because there's so many components that we have to keep track of, and there's multiple names that they're assigned, and how those individuals are distributed while still maintaining their familial crosses or their parental information versus their ID versus what ID they're given when they're given to a partner or ourselves is incredibly difficult, and we're working with Seafoundry to try to make that streamlined as possible by expanding upon hyperspectral imagery and really fine-tuning how we're going to capture that data. That would reduce the amount of time, the amount of people that are required in order to create this one ortho mosaic, from an image from the diver to a person processing it to someone actually analyzing it. That can all be streamlined.
Now, do we have sun for another 5 minutes to get that row, and that's basically 3/4 of this tank?
Dr. Jason Spadaro. I'm the program manager for coral reef restoration research at Mote Marine Laboratory.
I'm Scott Macdonald, the CEO and founder of Seafoundry.
Mote Marine Laboratory is a nonprofit organization dedicated to marine science, education, and restoration. We've been around since 1955, and here we're working on coral reef restoration and research, specifically on Florida's coral reef, but on coral reefs around the world.
We're a technology company dedicated to helping organizations like Mote do their work better, faster, stronger, and we've been working together for the last 2 years, I think now, on all sorts of things, but in this case they've been doing a lot of really great work on measuring the ecological success of their work at a sort of a larger scale than has been possible in the past, and that's where we saw an opportunity to see what Headwall could offer in terms of technology to help in that. What would be really great to see is different spectral signatures for non-restored and restored sites, and if possible, some of the species differentiation of what they've worked with in the past.
Developing a remote sensing capability that expands the scale at which we can do monitoring, and monitoring for predictive results is potentially transformative. Instead of using single species plots and focusing very heavily on genotypic level diversity within each species and creating these monocultures of single species outplant sites, we're moving more towards looking at classic ecological theory and using common garden approach and iterative outplanting on top of one another to drive ecological processes, and including succession, to try and facilitate higher order success.
If you were to count, you could see actually all 16 channels of the LiDAR. And it's flashing at 10 hertz, which is it's spinning at 60 RPM.
Pushing the field of marine restoration and conservation forward, but also, personally, giving our reefs a fighting chance of being there for the next generation of young scientists.
Seeing them have success in these efforts, they're close partners of ours, and our goals is really to see those reefs come back faster than we're able to do so currently, and be able to really differentiate between where we do take and don't take action.
Oh, like the plot location? Yes. Yeah, well, he knew that the previous outplants that we've done here were over here on the west end of the reef, so we wanted to make them disparate so that when you guys came, it was distinct. Yeah, glaringly obvious where we did not have living corals. If we're able to differentiate between live and dead coral, stressed and healthy coral, between species, potentially between genotypes within each species on the reef without getting in the water, and from an aerial platform, that dramatically increases the scope of what we can accomplish.
One of the wonderful things about hyperspectral imaging is, and particularly remote sensing, is that you can do things that may take an expert a lot of time to do, and you can do them automatically based on intrinsic properties of the things that you're looking at. And so in the case of the coral, I would hope that we'd be able to come up with some ways of automatically through imagery identifying species and genotypes of corals, and potentially being able to look at stress levels in corals and identify healthy versus unhealthy corals as part of this whole grand scheme of trying to restore damaged coral ecosystems. So he's going to start with a really quick flight to fly to the vertical register. It needs a very high rate of change of acceleration to get the filters out the accelerometers going.
We've got to peel out.
Yes, we can peel
Basically.
out, full stick forward, 2 or 3 seconds, full stick back, try to put the stop and rate of change of acceleration. And so once and it's doing that outside of the capture, probably run over there. And once he's got that done, then he's going to go to the first waypoint in his flight plan, set it to automatic, and then he's off the controls. So the drone is right here, and it's on flight line 1, 2, 3, 4. It's on flight line 6. That's about halfway through.
Yep, there it goes.
Preparation for field missions is really crucial. We were taking off and landing from a small area on a boat with a not the biggest, but also not a very small UAV. And to be honest, it was a little bit dangerous. We only decided to do that because me and Carson had done some practice back at the shop where we developed a confined space, and we said, what's the best way to go about this? Originally, we were going to practice just landing it in front of us, and then when we realized that, oh, well, we're landing right here and it's eye level, if somebody were to bump me and I hit the sticks a little bit or something, the drone could actually go flying towards us, and spinning blades at eye level is not ideal. And so even though in the end it looked more dangerous to catch the drone, we decided that that was actually the safest way to do it, and we practiced that. First we did it without a payload, and then we practiced it with a payload, and we decided we want to have a hard hat and gloves.
It started as a computer science major, and very rapidly decided that I didn't like that, so found my way to biological sciences, and then stayed on for a PhD in ecological sciences, and then meandered my way through the scientific community here in the Florida Keys, and ended up with Mote in this role.
The hardest part of the mission was catching the drone out of the air from the back of a pitching boat. Maybe more stressful than difficult, but it's an expensive system, and if it went in the water, it'd have been a total loss. And we worked together, and we were careful.
We worked at a scuba shop where we did certifications in a pond, even though I was from Colorado. I love the ocean, but didn't find my way to it until after some time in the industry working on manufacturing and products and next-gen technologies in that space. And trying to bring that here into this space has been a great privilege and honor.
Exploring these new and innovative technologies, especially underwater. I imagine just like we saw autonomous drone imagery being taken, there's certainly autonomous underwater vehicles that could do the same thing, but without that challenge of the surface of the water. Data capture is obviously changing as technology increases and advancements are made with things like hyperspectral imagery or robots and neural networks and AI. All of these things are changing the way that we capture data. But really the amazing bit is being able to integrate all of those different data streams into unified data systems that allow us to make predictions, react to changes, identify changes before symptoms are showing up on the reef. And that's where the partnership with Seafoundry and Headwall, wonderful data people over here and innovators. Bringing that into coral reef restoration is one of those things where it's synergistic, where you get more than just adding another group. It's this explosion in innovative use cases and technology development.
Something like Hyperspectral, where you can literally detect things that humans can't detect. That's a powerful new tool that we're just learning how to use.
We're still chunking through the data. It takes quite a while just due to the sheer size. I've been primarily looking at the flight data, and Carson's been primarily looking at the lab scans. And kind of the intention is we will analyze the lab scans first and then develop a model and then take that model and apply it to the remote sensing data. Carson has found that there are some strong peaks specifically around the 512 nanometer in the fluorescence region. We can pretty easily differentiate a couple of the different species of coral. Basically, we will finish our analysis on the tank scans and then apply it to the broader scene and say if we can detect anything or not. I think there's a strong application here. It's just finding the correct collection medium, really.
It's pretty special to see that kind of rig up in the air collecting data for this purpose, so way to go, Headwall.
Agreed. Thank you, Headwall, for coming out with us.
The most striking thing in the analysis that I've done so far, which is really very preliminary, is that there's some very strong fluorescence in the coral, and we can use those to differentiate amongst species. And I don't know that much about the species, but it looks like there's some very similar corals that look very similar to the eye, but have significantly different expressions of these fluorescent chemicals. And so I think that's going to be a part of figuring out which corals are which and potentially what is their health.
The sense of community and the ability that we're able to exponentially grow every year. For example, like with the hyperspectral imaging, with the Seafoundry partners, with different, like, for example, gene swaps for us, like we will produce larvae, we give some to partners, they give some to us, just to make sure that we are getting as much diversity back onto our reefs as possible has been so reassuring. And I know that the task at hand is very difficult and daunting, and sometimes I get kind of freaked out when I really think about it. But we have been able to produce resilient animals and tackle really large issues.
It looks like there could be a significant role for hyperspectral imaging in the process of culturing coral and analyzing the cultured coral in preparation for planting back into the environment.
If you want to donate money, that is huge. If you are here locally, we would love your help. We have a couple volunteers on site right now that are getting their hands wet, actually working with the coral. If that's not possible, if you live far away, like looking into what you can do in your local areas to help preserve and conserve like the natural resources that are there, because it all is all interconnected.
Ran out of batteries, so we're kind of done for the day, and it was 90, 95, and we all kind of decided to hop in the water, and Carson had a snorkel, and he went and he swam around, and he said, oh look, there's some big coral right under us. There's this, he found an interesting crab that was